Hello everybody, myself Dr. Icha Purak, retired university professor from Department of Botany, Ranchi Women's College, Ranchi. In this meeting, I shall try to explain some terms concerned with Mendel's experiment and laws of inheritance with the help of PowerPoint presentation. Now, in this is the first slide which shows the snap of scientist Gregor John Mendel, or he is also known as father of genetics. He tried to understand how characters are transmitted from one generation to other. For this, he performed experiments from 1857 to 1865 on a plant known as sweet pea or Pisum sativum. Now, genetics is the science dealing with transmission of characters from one generation to other. There are two aspects of genetics: heredity and variation. The characters which are inherited or in which the offsprings resemble to their parents, father and mother in some characters and also with each other, but at the same time differ in other characters. The similarities or resemblance between children and their parents and among children is known as heredity, and the difference as variation. The term heredity has its origin from Latin word heres or hereditatum, meaning higher or borrowed. Variation is the difference between children of same parents or differences between parents and children. Variations may be of two types: may be heritable, which are caused due to recombinations during meiosis. This recombination can be known as exchange of chromatid segments during crossing over, or non-heritable due to environmental changes, which can recover with return of favorable conditions. Transmission of characters over generations is controlled by genes. Gene is an inherited factor that determines a biological character of an organism. Gene, a segment of DNA, carries instructions for producing a specific trait in progeny or offsprings. The very first idea about gene came with Mendelian concept about factor or determiner, which he proposed on the basis of his experiment on garden pea from 1857 to 1865. He mentioned for transmission of characters over generations, something is transmitted which has a particulate nature. Mendelian factor is now termed as gene. He further stated that it exists in two alternative forms or alleles, and genes are present in linear fashion as beads on string. You can see in this diagram as beads on string on specific loci or position on chromosomes. Chromosomes are thread-like bodies present in nucleus of eukaryotic cells, plants and animals, and nucleoid of prokaryotic cells, bacteria, carry and transmit information in the form of genes. Number of chromosomes are fixed for a particular species or organism. Pisum sativum garden pea has 14 chromosomes diploid or 2n in zygote or somatic cell. Only 7 chromosomes, haploid or N, are present in male gamete and female gamete each. During fertilization, 7 chromosomes of male gamete fuse with 7 chromosomes of female gamete, resulting in formation of zygote having 14 chromosomes as 7 pairs of homologous chromosomes. Human or Homo sapiens have 23 pairs of chromosomes. Now, this slide shows the chromosomes of P, chromosome complement of P, which is 2n is equal to 14, and this P has 7 pairs of homologous chromosomes. The diploid number for this organism is 14, haploid number is 7, and locus is the specific position on a chromosome for placement of gene. The locus for flower color might be at chromosome number 5 at this position. Now, this diagram shows the 23 pairs of homologous chromosomes in Homo sapiens, that is human. Now, this slide again shows the homologous pair of chromosome, and as I have already explained that genes are present in linear fashion on the chromosome as beads on string, and for a particular trait chromosomes are present at particular position which is known as locus, gene locus. Now, for example, the gene for character height is can be symbolized as T dash, and its two alternative forms or alleles as capital T and small t. Capital T is responsible for alternative form which is known as tallness, and small t is responsible for dwarfness. Tall and dwarf are allelomorphs or phenotypes. Example is tall trait height of plant may be tall versus dwarf. In the same way, trait color of flower may be purple versus white. It is not always necessary that a gene may have only two alternative forms or alleles. Sometimes a gene may have two or more alternative forms or alleles, but only two of them are found within individual or a cell at a time. When there are more than two alleles for a gene, these are known as multiple alleles, as for example, blood types in human, which is A, B, AB, and O. Now, the experiment was hybridization. Now, let us know what is hybridization in plant. Hybridization is mixing up and is a technique to improve the varieties both in animal and plant. But today we will see it for plants. Hybridization is frequently employed in agriculture to make stronger, healthier plants with desirable characteristics. In plants, hybridization is done by artificial cross pollination. For example, a cross between parent 1 tall with the genetic makeup or genotype capital T and parent 2 dwarf with the genotype small t produces progeny with genetic makeup capital T small t, which is a hybrid. hybrid or filial generation or F1 generation. Hybridization was the basis of Mendel's historic experiment with garden peas. Inheritance studies require crossing plants with contrasting or alternative traits by considering one plant as male and other as female, although both parents are bisexual plants. दोनों ही पौधे बाइसेक्सुअल हैं, लेकिन एक पौधा जिसका हम पॉलन ग्रेन यूज करते हैं उसे मेल प्लांट कहते हैं, और जिस पौधे का कार्पेल यूज करते हैं उसे फीमेल प्लांट कहते हैं. Now this slide shows the process of hybridization. Pea plants or Pisum sativum are naturally self-pollinating plants and so can be easily used for artificial cross fertilization or hybridization. In this, emasculation is a process which is removal of male reproductive parts. Before anthesis, the anthers are removed and it is followed by dusting stigma or carpel with pollen from other flowers. In this diagram, you can see the first step, that is removed stamens from purple flower. Then in the second step, the transferred pollen from stamen of white flowers to carpel of purple flowers. In the third step, pollinated carpel were left to mature into pod, and in the fourth step, planted seeds from the pod. Now, what are homozygous and heterozygous? Homo means similar, hetero means different. During sexual reproduction, haploid gametes fertilize haploid female gametes to produce diploid zygote having two alleles present on two homologous chromosomes together. Haploid gametes have only one allele of a trait and are produced by meiosis or reduction division. There are two types of cell division, mitosis and meiosis. Mitosis is known as homotypic division and meiosis is known as reduction division. In mitosis, the number of chromosomes in the daughter cell remain the same as the parent cell, but in meiosis, the number of chromosomes in the daughter cells become half or reduced to half in comparison to parent cell. If parent cell is having 14 chromosomes, the two daughter cells will have only 7 and 7 chromosomes. During prophase 1 of meiosis, crossing over, exchange of segments between non-sister chromatids of homologous chromosomes take place, and so after fertilization, different combination of alleles are resulted in different gametes, and so in zygote or offsprings. If the two alleles in a zygote are identical or are of same nature, combination is called as homozygous. As for example, capital T, both on both the homologous chromosome, allele capital T is present. Both are dominant alleles, and if on both the chromosomes small t recessive alleles are present, it will be known as homozygous. Homozygous are also known as pure or true breeding because they will give the same type of plant. If in zygote two alleles are different, not identical, as for example, capital T small t is known as heterozygous combination or impure or false breeding because they can give two type of gametes, one having capital T, other having small t. Now, what is genotype and what is phenotype? Each gene has two alternative forms or alleles control expression of a character or trait having two phenotypes or allele or more. Genotype is the specific allelic combination for a certain gene or set of genes. Genotype of pure tall plant is capital T, capital T. Genotype of hybrid plant is capital T, small t. Genotype of pure dwarf plant is small t, small t. Phenotype literally means the form that is shown. It is outward, morphological appearance of a particular trait. Mendel's pea plant exhibited the following phenotypes. Tall and dwarf plant for the phenotype height, yellow and green seed color phenotype, purple and white for the flower color phenotype. Now let us discuss about dominant and recessive alleles. Dominant जो अपना प्रभाव दूसरे पे डालता है और रिसेसिव जो दूसरे से प्रभावित हो जाता है, ये यहाँ हम देखेंगे। आम जिंदगी में भी हम लोग देखते हैं कि जब हमसे ज्यादा कोई किसी चीज में माहिर होता है तो हम अपने में जो है उसको शो नहीं कर पाते। डोमिनेंट in a dominant can be explained as in a cross between two alternative forms of a trait, an allele that expresses itself at the expense of other alternative allele in F1 generation. The phenotype that is expressed in F1 first filial or hybrid generation from the cross of two pure lines is dominant phenotype. As for example, in a cross between a pure tall and pure dwarf plant, all plants of F1 generation were tall. It means allele capital T dominates the expression of allele small t. Recessive, in a cross between two alternative forms of a trait, an allele whose expression is suppressed in the presence of a dominant allele, the phenotype that disappears in the F1 generation from the cross of two pure lines and reappear in the F2 generation. Dwarfness or recessive did not appear in F1 generation because allelic combination was capital T small t. In presence of allele capital T, allele small t was not able to express itself as it was suppressed. But in F2 generation, when allelic pair combination was small t, dwarfness appeared. Now, what is monohybrid, what is dihybrid, what is trihybrid and polyhybrid crosses, and what are its ratios? So, what do we mean by monohybrid? Whenever such a cross is done or artificial cross pollination is performed, the male gamete is transferred from other plant to the carpel of other plant. Then whatever characters the pollen is possessing will be transferred. But what we are considering makes it monohybrid or dihybrid or trihybrid cross. Monohybrid cross, Mendel performed crosses for all the 7 traits. During recording results of hybridization, he focused only on one trait and its two alternative forms at a time. Such cross is known as monohybrid cross. For example, cross between a tall male plant and dwarf female plant, trait height of the plant, F1 in all tall, F2 3:1 tall or dwarf is known as monohybrid phenotypic ratio. What he observed that in F1 all the plants were tall. Although he was expecting that the plant will be of intermediate height. Tall plant is about 2 meter long and dwarf plant is about 1.5 meter long. And he was expecting the hybrid will be in between plants were tall. Means only one allele, that is capital T, expressed itself and the other allele did not express itself. And when we consider only one trait at a time, it is known as monohybrid cross. Similarly, when at a time two traits, height of the plant and color of flower, are considered, it is known as dihybrid cross, having 9 is to 3 is to 3 is to 1 as dihybrid phenotypic ratio. In this 9, out of 16 combinations, 9 combinations are tall plants with red flowers, 3 plants are tall with white flowers, 3 plants are dwarf plants with red flowers, and 1 is dwarf plant with white flowers. When 3 traits are considered at a time, it is trihybrid cross, trihybrid phenotypic ratio, having 64 combinations, 8 type of male gametes and 8 type of female gametes, will be 27 is to 9 is to 9 is to 9 is to 3 is to 3 is to 3 is to 1, having different forms, having all the 3 dominant, 2 dominant and 1 recessive, 1 dominant and 2 recessive, and all the 3 recessive alleles. Now let us also know about reciprocal cross. Means we can take tall plant as male or tall plant as female. In the same way, dwarf plant as female or male parent. For example, if in a set of experiment tall plant is used as male parent and dwarf plant as female parent, in its reciprocal cross, tall plant is used as female parent and dwarf plant as male parent. In both cases, results are the same as all F1 plants will be tall and F2 generation will show tall and dwarf plants in 3:1 ratio. Now, I think I have been able to tell about the terms which are related with Mendelian experiment and laws of inheritance. We can also see it with the help of whiteboard. Now this whiteboard helps us to write some of the important terms like what is genetics. We have discussed genetics. Then genetics is the science which deals with transmission of characters from one generation to other. Then we have come to know what is heredity. What is heredity? Heredity is the similarity between parents and offspring and among the offspring. Variations are the differences between parents and offspring and also among the offsprings, and variations can be heritable or non-heritable. Then we have also discussed about gene. What is gene? Gene, now we know that it is the carrier of heredity characters from one generation to other, and it carries all the information for expression of the gene. But gene was for the first time known as factor or determinant and was given by Professor Grigor John Mendel, who was working on hybridization experiment. Now, along with that, we should also know that how, where the genes are present, which are the carriers of genes. Carriers of genes are chromosomes. Chromosomes are thread-like structures chromosomes are thread-like structures and are present as homologous pairs on the chromosomes. The genes are present as beads in the string, and for position of genes for a trait, the position is fixed, which is known as locus. The gene for character of height is can be symbolized as T dash, which has two alternative forms, capital T and small t. Capital T is responsible for tallness and small t for dwarfness. Capital T and small t are known as alleles. And tall and dwarf are known as allelomorph or phenotype. Allelomorph or phenotype. The chromosomes for an organism, the number of chromosomes for an organism are fixed. There are 23 pairs of chromosomes in Homo sapiens, 7 pairs of chromosomes in Bison sativum or switch pea. The alleles on the two chromosomes may be alike. If they are alike, they are known as homozygous. For example, capital T, and if it is small t, then also it is homozygous. But if they differ, they are known as heterozygous. That is capital T, small t. Homozygous are pure breeding, whereas heterozygous are false breeding. Then we have also come to know about what is genotype and what is phenotype. The gene combination is known as genotype and the physical appearance is known as phenotype. Now, what are monohybrid crosses? Monohybrid cross. What is hybridization? Hybridization is the artificial cross pollination. Monohybrid cross is the cross with two pure parental plants when only one of the trait and its two alternative forms are considered. In the same way, there is a dihybrid cross, which at a time we can consider two characters and its alternative traits, their alternative traits. If three characters are considered at a time, it will be known as trihybrid cross. Then there is a reciprocal cross. If in the first set of experiment we have taken tall plant as male plant and dwarf male as the dwarf plant. So with this, we have been able to know the terminology concerned with Mendelian experiment and laws of inheritance. I think it is enough. So for today's meeting, and with your permission, I want to end this meeting. In the next class, I'll explain. In the next meeting, I'll explain what is monohybrid growth and what are the laws based on the monohybrid growth. And in subsequent meeting, we will discuss dihybrid growth.